Good day, this is Professor Wills, CBMD PhD, and today is Monday, June 6th, 2022. It is 5:18 and 35 seconds, AM Eastern Standard Time, and today's research, breast cancer and diabetes. So let's hop to it here. The Apparent Perils of Breast Cancer and Diabetes, date May 30th, 2022. Source, University of California, San Diego. Researchers have discovered a mechanism linking breast cancer and diabetes, each of which promotes development and growth of the other. Breast cancer and type 2 diabetes would seem to be distinctively different diseases, with commonality only in their commonality. Breast cancer is the second most diagnosed malignancy after some types of skin cancer. Approximately 1 in 8 U.S. women will develop invasive breast cancer over the course of their lifetime. More than 10% of the U.S. population has diabetes, with an estimated 2 in 5 Americans expected to develop the chronic disease during their lifetime. However, past research has uncovered associations between the two diseases. Women with diabetes, for example, have a 20 to 27% increased risk of developing breast cancer. Insulin resistance, a key characteristic of diabetes, has been associated with breast cancer incidence and poor survival. Population studies suggest diabetes risk begins to increase 2 years after a breast cancer diagnosis, and by 10 years post-diagnosis, the risk is 20% higher in breast cancer survivors than in age-matched women without breast cancer. But these epidemiological linkages are not clear-cut or definitive, and some studies have found no associations at all. Researchers describe a possible biological mechanism connecting the two diseases, in which breast cancer suppresses the production of insulin, resulting in diabetes, and the impairment of blood sugar control promotes tumor growth. No disease is an island, because no cell lives alone. Researchers describe how breast cancer cells impair the function of pancreatic islets to make them produce less insulin than needed, leading to higher blood glucose levels in breast cancer patients compared to females without cancer. The culprits, according to researchers, are extracellular vessels, hollow smears, secreted or shed by cells that transport DNA, RNA proteins, fats and other materials between cells, a sort of cargo communication system. In this case, the cancer cells were found to be secreting microRNA 122 into the vessels. When vessels reach the pancreas, they can enter the islet cells responsible for insulin production, dispense their micro 122 cargo, and damage the islet's critical function in maintaining a normal blood glucose level. The cancer cells have a sweet tooth. They use more glucose than healthy cells in order to fuel tumor growth, and this has been the basis for position emission tomography scans and cancer detection. By increasing blood glucose that can be easily used by cancer cells, breast tumors make their own favorite food, and meanwhile deprive the essential nutrient from normal cells. The research was conducted using mouse models, which found that slow-releasing insulin pellets, or glucose-lowering drug, known as the SGLT2 inhibitor, restored normal control of glucose in the presence of a breast tumor, which in turn suppressed the tumor's growth. And these findings support a greater need for diabetes screening and prevention among breast cancer patients and survivors. Noting that an inhibitor of micro 122 is currently in clinical trial as a potential treatment for chronic hepatitis C. As we already talked about, it has been found to be effective in restoring normal insulin production and suppressing tumor growth in mouse models of breast cancer. And these micro 122 inhibitors, which happen to be the first microRNA-based drugs to enter clinical trials, might have a new use in breast cancer therapy. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoy listening. Until next time, have a great day.